For today's class, we will be doing a lot of drawing. So if you remember, we have four major macromolecules. So we have the carbohydrates, which other ones enough? Macromolecules. Carbohydrates, any other?
Protein.
Protein. Two more. All right, the next one is fats. Right, that's correct. Fats. And nucleic acid. All right, but for this topic, we won't be looking at nucleic acid. It's carbohydrate, protein, and fats. So these are the three macronutrients we'll be looking at. For this class, we're going to focus on the different drawings and reaction we will have to do for each of them. So we're going to start off now with carbohydrate. All right, now on the carbohydrates, we're going to be looking at the structure of glucose, fructose, sucrose, and these are our sugars. Glucose, fructose, sucrose. We're going to be looking at glycogen. We're going to be looking at starch. And we're going to be looking at cellulose. Put it on here. Right, so these sugars, right, we can classify them as simple or complex, right? Now, glucose, it is a simple sugar. Do you know the general term that we give for a simple sugar? Start with an M. Right, I will remind you. So simple. These are more complex. Right.
Is it like again?
Repeat.
Macronutrients or something like that?
Repeat.
Macronutrients.
Oh, not in this exact case. All right, hold on, let me start it off. It's mono.
Oh, mono.
Yeah, that word. Mono. Saccharide. Are you hearing me clearly or is it breaking up? Let me know.
Clearly.
All right. Right, so it is monosaccharide. Mono means one. So all of these are sugars, right? The simple one is made up of one structure. I'm going to be more specific a little, all right? The term saccharide is referring to sugar. Mono, it's one sugar unit. So monosaccharide, we're basically saying one unit of sugar. Fructose is also a monosaccharide. Sucrose is a disaccharide. With this one, it has 2 sugar units. And glycogen polysaccharide. So glucose and fructose, those are the 2 monosaccharides that you need to know. All right. Sucrose is a disaccharide. You will have to know the structure of sucrose. But the next one, they don't need to know to draw it. You have maltose. So our next example of disaccharide is maltose. Maltose, lactose, lactose, and galactose. These are some common ones, right? But in terms of knowing to draw the structure, is only sucrose. All of these here, you must be able to draw them. But the first thing we're doing is just classifying them. So glucose and fructose, monosaccharide, 1 sugar unit, disaccharide, 2 units. The complex one now, which is the polysaccharide. Remember in organic chemistry, polymers, so polysaccharide meaning many sugar units, not 1, 2, or 3, a lot. Right? So many. So that is what we are going to be dealing with. We need to know to draw each of them. I'll give a minute to just take what you need from the board and then we continue. So just let me know when I can share the board. All right, so let's focus now on glucose. So as you know, this is at the Cape level, so a lot more detail is required of us. The first thing we need to know is to draw glucose. Glucose is an hexose sugar, right? Hexose mean it has 6 carbons. If glucose is a carbohydrate, which elements do you think should make it up?
Repeat that, sir.
So glucose is classified as a carbohydrate, right? So do you remember the elements that are present in carbohydrates? Apart from carbon, do you remember which other elements are present?
Hydrogen.
Hydrogen, that's correct. One more. So
Oxygen.
right. So it consists of carbon, hydrogen, and oxygen. Do you remember the formula of it?
Of glucose,
Yeah,
C6H12O6.
right, that is correct. Now at the Cape level, we are not just interested in the molecular formula here. We're interested in the structural formula, all right? So how does the 6 carbon, the 12 hydrogen, and the 6 oxygen, how are they actually connected to each other? We're going to look at the arrangement. For glucose, I'm going to start with a carbon, right, at the top, connected to a next carbon. So these lines, what are these lines representing? This line is what? Do you know what it is representing?
Bond, sir.
That is correct, right? So it's a bond. So this carbon is bonded to this one. We have an oxygen there. Sorry, getting ahead. So this part is like a hexagon. 1, 2, 3, 4, 5, 6-sided figure, right? With a carbon on top. Now, as we know, carbon atoms always form 4 bonds, right? So that means this carbon on top, it needs 4 more bonds. 2 of it is hydrogen, and the next one is an OH group. All right. If you look at this carbon, it has 1, 2, 3 bonds. Something is missing. That is a hydrogen. This carbon here, it must have hydrogen and OH on it. So for this one, we make the hydrogens, each atom attached, we point it up and down. So if the hydrogen is going up, I'm going to make the OH group point downwards. Before I go any further, this carbon here, this is carbon number 1. Right? So that's carbon number 1. The OH group is pointing down. When it is pointing down, we classify it as alpha. So I'm drawing the structure for alpha glucose. So that's the symbol alpha, as you know. Right? This is alpha glucose. The reason we're classifying it as alpha is because the OH group on carbon number 1 is pointing downwards. If it is pointing upwards, we're going to give it a different symbol, which is beta. All right. So I will draw the beta afterwards. Let's continue. And this, what we're drawing here, it's the ring structure. All right. Ring structure. They have an open chain and they have the ring structure. We start with the ring and then I'm going to do the open chain. All of this you should be able to do from memory. As you get it, you start practicing. The second carbon, the OH group, is pointing outside. So all we're adding is H and OH to each carbon. This is carbon number 2. This is carbon number 3. So we start numbering from this carbon. 1, 2, 3, 4. This is carbon 5, and this is carbon 6. On carbon number 3, the OH group is on the inside of the ring. The hydrogen is on the outside. And then we have an OH pointing down and the hydrogen pointing up. Right? And that concludes the structure of alpha glucose. Right? Now, we won't be using this detailed structure where you join out every carbon and every oxygen. It's the first time we're doing it. That is why I'm showing you every carbon. In most cases, all you need to do is this. Now, where 2 lines meet, I'm just highlighting it. Those are where the carbon atoms are. Wherever you see 2 lines connecting, your carbon atoms are present there. So carbon 1, right? Look over here now. The end of this line is meeting the end of this one. So we have a connection here, 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 and here. And the next carbon now, put on top. You don't have to draw out the H's. You can simply put CH2OH. So we condense it. Instead of drawing out the two hydrogens, we just write CH2OH. All right. And then you would continue now, put on the OH and the H's. So basically this part is just practice. All it takes is practice. And you will definitely remember how to do it. All right, so this is still alpha glucose. All right. Beta is, everything is going to be the same for beta with one exception. And if you compare the two structures, everything is the same except for the position of the OH group on carbon number 1. When it is pointing upwards, that is beta glucose. When it is pointing downwards, it is alpha. That is the only difference between your alpha and beta glucose. And carbon number 1, so remember this right here, that is carbon number 1. All right, carbon number 1, let me bring down this. Carbon number 1, it is called your anomeric carbon. ANOMERIC. Anomeric carbon atom. All right, and that's simply the carbon atom where the OH group can change its position. You'll notice every other OH group are in the same positions, but the OH group on carbon 1, when it arranges itself in space, it can either be pointing up or down. So it is the carbon atom. So when we say anomeric carbon atom, All right, so it can arrange itself differently, up or down. All right, so as I said earlier, what we're doing here is the ring structure. We also have the open chain. Open chain look like this. And so up here, this is carbon number 1, and this is carbon number 6. So the COH with the 2H, all of this, you could have written it as CH2OH, all right? And the C double bond O with the H, that could have been written as CHO. You could start CHO with 1, 2, and continue until you get to carbon number 6. You put CH2OH. But I'm drawing out all of the bonds so you can see. All right. The important thing to note here is the position of the OH group on carbon number 3. So this is carbon number 3. Notice every other OH group is pointing to the right, but on carbon number 3, it is pointing to the left. When you come to the ring structure, you notice all the OH groups are pointing outside of the ring, but the one on carbon number 3 is pointing on the inside. So when this open chain is converted into the ring format, the OH is pointing inwards. So whenever, just a side note, when glucose is in solution, you can have the mixture of the open and ring format. So it can convert itself into the ring. All right. And it can switch between alpha and beta. So you should be able to draw the ring structure for alpha and beta and draw the straight chain as well. The next thing I want to highlight, this group here, CHO. That is an aldehyde group. All right. From chemistry, it is an aldehyde group. Let me see if I have anywhere I can write. All right. So because it has an aldehyde group, glucose is referred to as an aldose sugar. All right. So it is also an aldose sugar because it has the aldehyde group. And that is also why it is a reducing sugar. We will get into this in next class when we're looking at the actual details. So for this class, basically focusing on the structure. Next week, now we do some discussion on how the structure affects its properties. Right? But I'm just telling them now that is why it is a reducing sugar because of the aldehyde group present. Right, I'm going to pause here and let you So fructose, it is also an hexose sugar. It is also called, remember, glucose is called aldose because it has an aldehyde group, while fructose is called a ketose because it has a ketone group. All right? So glucose is an aldose sugar, but fructose is a ketose sugar because it contains a ketone group. All right? For this, we're going to draw just the ring structure. It is the ring structure. Well, let me show you the two of them because I need to show you the ketone group. Now, remember, glucose, right? Start off with CHO. That is the C double bond OH. It starts off with the aldehyde group. Fructose doesn't start like that. It starts with CH2OH. Then you have carbon, double bond, oxygen. Notice again on carbon 3, OH is on this side. It is kind of similar to the structure of glucose in a way. 1, 2, 3, 4, 5. So the open chain of fructose, the starting and the ending, it has two CH2OH group. What makes it a ketone? Let me get the next ink. This right, so ketone, C, double bond O, C. So the carbon with a double bond oxygen, two carbons are also attached to it. All right, so bond. All right, so that is the ketone group. So that is what makes your fructose a ketose sugar because it has a ketone group in it. All right. Now, in terms of the ring structure, now it's a little different from glucose. So it's actually shaped like this. All right. Now, this is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4. If you remember, when we did glucose, I want to point out something so you don't make that mistake. When we did glucose, right, the OH group was here because this was carbon 1, this was carbon 2, and this was carbon 3. But bearing in mind of a fructose, because this is carbon 1, carbon 3 is actually here. So this OH group will be inside the ring, and this one here will be outside of the ring. All right. So with glucose, this OH, it will be outside of the ring because it's actually carbon number 2, and this is 3. With fructose, this is 3 and this is 4. All right. Just bear that in mind where the OH groups are. All right. So let's continue. This is carbon 5, Carbon 6 is up here. Now carbon 5, it's actually a hydrogen that will be here, not a OH. All right. So unlike glucose, we have OH here. And if you check, right, this is carbon number 4 on glucose. This is carbon 5. If you check carbon 5 on glucose, it is CH2OH, right? This right here is carbon 5. It has a hydrogen and then CH2 above it. So it's just the sequence of numbering will make it look different. So the shape changed the arrangement, but in terms of connection is the same. This is carbon 5, CH2 above it, hydrogen attached to it. And glucose, this is carbon 5, CH2OH above it, hydrogen attached to it. All right. So it's just paying careful attention where to put what exactly. So not because you have OH here, mean you're going to put OH here. There's no OH on carbon 5. There's no OH on carbon 5. All right, so just bear that in mind. So it would be carbon 4, carbon 5. This is carbon 6. All right. The OH group is pointing downwards. All right. So that will give us beta. Sorry. If it's pointing downward, it is alpha. When the OH is pointing upwards, then it is going to be beta. So this is the beta form of fructose. So those are the structure for fructose that you should know. But the only one you really need is the beta one, right? Because we're going to use it to draw sucrose. Right, so just take off the structure and then we continue. Right, so for the disaccharide, we only need sucrose. You don't need to know the lactose or the maltose. Earlier I said that galactose is a monosaccharide. I think I wrote it as a disaccharide. Galactose is a monosaccharide. I can put it under monosaccharide. All right. All right, so sucrose, it is made up of alpha glucose. So once you get to the disaccharides and polysaccharides, you need to know the monosaccharide that make them up. So for sucrose, it is alpha glucose and beta fructose. All right. Now these reactions, they are formed by condensation reactions. So the type of reaction is taking place to form sucrose. It is a condensation reaction. Now, in this type of reaction, two molecules are joined together with the removal or if you want to say elimination and that small molecule in most well in all of for our purposes it's always going to be the water. With condensation we are joining two molecules by removing a molecule of water in the process. All right. The opposite, I'm just going to put it here and you make a note, all right? It won't just go for sucrose, so it's just, all right, and put NB. The opposite they have condensation and they have hydrolysis. So to form sucrose, we're going to join up glucose and fructose by removing a molecule of water. And so that is condensation. Now, if you want to break up back, if you want to break sucrose, you're going to add back water to it. So in hydrolysis, continue. In hydrolysis, again, for our purposes, this small molecule is water. So basically, one is condensation. So it's like a reverse reaction. All right, so after you've finished here, I'm going to draw the actual structures and show the reaction. Containing sucrose is also a non-reducing sugar. And I will show you why in a few. All right, so let's go now. So we said it is made from alpha glucose. So let's draw alpha glucose. Remember alpha, it points downwards. Now with sucrose, right? All right, let me do this short quickly on this side. If you remember carbon number 5, right? It has a hydrogen attached to it. Now the reaction that is taking place is when sugar molecules are going to react. OH groups from each sugar are going to take part. So remember we said that what? We're going to join them up by removing a molecule of water. So what is supposed to happen? OH group from one of the sugars react with OH group from a next sugar. So we take OH group from one of them. Actually, let me just draw the carbon bond. We'd have a C bonded to a OH. Same thing here, right? And then you take OH from one of them and hydrogen from the next one, right? And so you'd have C bonded to this O, which is attached to this C. So if you look at fructose, right? If we should take it up and put it here, you will realize that there is no OH for it to bond to, right? You have OH and you have H. But it's supposed to be two OH facing each other. Now, on carbon 2, so this would be carbon 2, right? and this would be carbon 1. We have our OH group here that is available to react, but it is on this side, right? So when we're going to show the reaction, we have to flip the molecule around. We have to make this OH in line with this one, all right? So even though fructose is like this, it has to be arranged so that the two OH groups, the carbon 1 OH group is in line with carbon 2 OH groups, all right? So it's because of, for that reason now, when I'm drawing sucrose, I will try to get some organic chemistry models so that you can actually see this in 3D, right? It will be better because even if we draw it in 2D here flat, it would not actually be, it will not actually be flat, right? So when I do the rotation, you will understand better. But in terms of drawing, this is what it will look like. So the shape of it is going to stay the same, which is this, all right? Now, this right here, this is not carbon number 5. Remember, we flip it around. This is now carbon 2. This over here is carbon 5. So when you're doing sucrose, just know that the fructose molecule will flip it around. So this is carbon 2, all right? And so you have your OH here and the CH2 OH here. All right? Let me erase the number 5. CH2 OH. If this is carbon 1, this is carbon 2. I mean, this is carbon 3. So the OH and this should be inside of the ring. This is carbon 4. All right, on the outside. You can put on your hydrogens. All right, so all you're going to do now, take off OH from one of them and H from the next one. So for this reaction, it is carbon number 1 from glucose and carbon number 2 from fructose. So we also have to know which carbon atoms are taking part in the reaction, or we should say the OH group on the particular carbon atom. It's the OH group on carbon number 1 for glucose and the OH group on carbon number 2 for fructose. All right, so let me just erase. You have to be able to show this reaction. They can ask you to show it. So after you do this, you draw your arrow. We're getting rid of water. So we're eliminating H2 and the O. What is left now? Let me switch colors. CH2OH. So this glucose here, right? This carbon one will be joined to this oxygen.
There's another for me? No. Pwede just take it capture
Write it down here.
put it in your hand.
Then you just put on back everything else. All right, so that is fructose. Sorry, sucrose. And the type of bond is a 1,2 glycosidic bond. So this bond right here, that's your glycosidic bond. Every sugar bond is called a glycosidic bond. Or you might see link. It's called a bond or a link. So 1,2, the reason why it is 1,2 is because it's carbon 1 and carbon 2. So it's a 1,2 glycosidic bond. So you should be able to show this reaction. While you write, in terms of the food test labs that you do, right? You know that, as we say, sucrose is a non-reducing sugar. But remember now, glucose and fructose, they are both reducing sugars. But this bond that is formed right here, it prevents any reaction from happening, right? So what happens, if you look back at the method, we normally add acid first. So when you're doing non-reducing sugar test, we add acid to it. So with reducing sugar, you would add basic solution and then place it in the hot water bath. But for the non-reducing sugar, the first thing we do is add acid to the sugar and put it in the hot water bath for a few minutes, a minute or so, right? And while we're doing that, we're hydrolyzing the sugar. Remember, condensation, we're joining two compounds, right? By removing the molecule of water. So with the hydrolysis now, we are breaking this bond. If you break this bond here, we are going to break it and we're adding back the OH to this one and the H to this one. So what happens, you start out with a non-reducing sugar, but you are ending up with reducing sugars. So how the process works, right? You start out with non-reducing sugar. You add the acid. The acid splits up the components of the non-reducing sugar, which is break up glucose and fructose. Glucose and fructose on their own are reducing sugars. When you boil it in the acid, and then you take it out back and add a little base to neutralize the mixture, then add Benedict solution. You see, when you add the Benedict solution after adding the acid, there is no longer non-reducing sugar. in the test tube. You only have reducing sugar because they already carried out hydrolysis. All right. Now, how you confirm it's the non-reducing sugar is to just add Benedict's solution directly to it. You will not get a color change. So adding Benedict's solution directly to sucrose, you will not get a color change because the carbon one here, it's not free up. This bond here prevents any reaction from occurring. All right, so if you just add it directly, no reaction occurs. But if you add acid first, the acid will break this glycosidic bond. You end up back with the glucose and your fructose. And so when you add Benedict solution afterwards now, they will reduce the copper 2 ions present to copper 1. Understand? Yes, no?
No, sir.
You want to repeat? Sure, not a problem. All right. So you're reducing sugar, right? Which is your glucose and fructose. Glucose have your aldehyde group and the fructose as a ketone group. They will react with the copper 2 plus in Benedict's solution. All right. Let me just double check to see if I'm recording. Yes, all right. Right, so these two are reducing sugars. When you add copper 2 ions to them in the form of Benedict solution, right? Copper 2 plus gets reduced to copper 1 because the reducing groups, which is the aldehyde group and the ketone group, are available to do this reaction. As you can see here, sorry. As you can see here, the carbon 1, right, with carbon 2, which is this linkage here, this linkage, the groups, the OH group that would have otherwise partaken this reaction here is not available. It is this link that makes it the non-reducing sugar. The aldehyde groups and ketone groups are not there. So with this link, no reaction is possible. Copper 2 ions will not be reduced to copper 1, right? When you add the hydrochloric acid to sucrose, right? So let's say you have your beaker with your water and you apply heat, you apply your heat and you get your test tube now. Just happening here, just a second. You add your test tube to it. I'm going to put RS for reducing, sorry, non-reducing. I'm going to put S for your sugar, right? So you add your sugar, and then you add some acid to it. You add the acid, the sugar, and you boil it, right? When you boil the sugar in the acid, this happens. The reverse reaction happens. So before we add sucrose present in the solution, when you add the acid to it, sucrose is converted to glucose and fructose. Are we clear on this part? Adding the acid hydrolyzes the glycosidic bond. When I say hydrolyze, break that, put OH here. All right. This already had the H, the O, I'm putting none but the H on it. That is what happens when we add acid to the sugar. So question, If this happens, right, do we still have sucrose in the test tube? It's sucrose in the test tube. Okay, sorry about that, it went down. Nadiv, a side message. It is being recorded, so you can watch it afterwards. Right, but we lost the screen, so I have to redo it. Right, but the point I was making, right? I'm just going to use a simplified diagram to explain it. So let us say this is glucose, right? And this is fructose. On their own, separately, they can reduce Benedict's solution, right? On their own. But if we join up the two of them, once they are joined together, so now we have sucrose, the reaction that occurs when they join up, it prevents, basically they lose their reducing ability, put it that way. So on their own, they have their reducing ability. All right. Once you join them up, they lose their reducing ability. All right. And so because they lose their reducing ability, we say it is a non-reducing sugar. So sucrose is a non-reducing sugar. In the non-reducing sugar test, if we add Benedict's solution to sucrose, nothing will happen because it has lost its reducing ability. So glucose and fructose, when they join to form sucrose, they lose their reducing ability. So adding Benedict's solution to this does nothing. So the Benedict solution will stay blue, right? However, if I should add acid now, if I should add acid to sucrose, what is going to happen? It breaks the glycosidic bond. So if the glycosidic bond is broken, my question is, do we still have sucrose or we have glucose and fructose? Which is it?
Glucose and fructose.
Exactly. So now, after we add the acid, we go ahead now. So first we add the acid to break the bond. Then we go ahead and add Benedict's solution, right? So if we add acid first, which we know will break the bond, giving us back glucose and fructose, when we go ahead now in the second part to add Benedict's solution, are we adding Benedict's solution to sucrose, or is it now glucose and fructose?
It's going to be added to glucose and fructose because now it will be testing for reducing sugar.
Please repeat the last part.
Because the glucose and fructose are now reducing sugar, the Benedict solution will be able to test it.
Right. Because now we have two reducing sugars present. So you start out with a non-reducing sugar, which is sucrose. But by the time you add the Benedict solution, you have two reducing sugars present. All right. So that is why, so you can explain your food test now. So with the non-reducing sugar, when you start out, it is a non-reducing sugar. But once you add the acid to it, the glycosidic bond is going to be broken. And once you break that bond, you no longer have your non-reducing sugar. You have your reducing sugars. And that is why you will get the color change. All right, so when we do it, I know how it is said in the lab, you might think that you're actually testing for, like you're actually carrying out a reaction with the non-reducing sugar. But in reality, when you add the Benedict's solution after the acid, it's not a non-reducing sugar that is giving you the color change. It's actually the reducing sugars that made up the non-reducing sugar that is there. All right. So that is what happens in the reducing and non-reducing sugar food test. All right. So we did sucrose, that's the first one. Let us do cellulose. All right. All right. So I'm going to clear the screen. That's okay, right?
1 second, sir.
Okay, not a problem.
Okay, go ahead.
All right. And so cellulose is made up of beta glucose. The type of bond is 1,4. As I said, any sugar bond is called glycosidic bonds. All right, so let's start. I'm going to leave off the OH to keep it simplified. So I'm just going to work with the OH and carbon 1. Remember it's beta, so on carbon 1, the OH is pointing up. Now the thing with cellulose, the molecules are going to flip one after the other. I would say rotate 180 degrees. So if this one is pointing up, the next one beside it must be pointing downwards. So if you want to know how to do it, right, just draw back this shape here. And, before I do that, so this shape here, right, on the next diagram, the CH2OH, it will come here. All right. CH2OH, why is this not writing? It will come here. The oxygen, it will come here. All right. So let's check now. I'm drawing it back. All right. But I said that the oxygen for this one, it must come here. So I put it there. All right. And the CH2 that was here, it comes here. All right. Hold on. CH2, OH. All right. Let me put on the OH and carbon 4 as well, because as we said, the reaction is between carbon 1 and 4. Let me erase this and this. All right, so carbon 6, how do you know to number it? All right, carbon 1 is always next to this oxygen. So here's the oxygen, that means this is carbon 1. This would be carbon 2, 3, 4, 5, 6. All right. So carbon 1, and you just go around. Just a second. Let me move the plus sign. I should have curled it up a little so you could see it. Oh, next thing, look here. When you flip this over, right, this OH group would come up here. Let me use the next ink. Sorry. So when you flip this over, the OH group come up here and the hydrogen come down here. That's for carbon number 4. So you would have the OH pointing up. Just a second. H. So you have the OH pointing up and the H is pointing down. All right. Again, it's a condensation reaction. So you can take OH from whichever one you want. All right. You take OH and H from the next one. This is carbon 1, right? And this is carbon 4. So carbon 1 is going to be bonded to this oxygen. All right. Let me do a next one. So let me just put the plus sign in between because it's a reaction. So this is reacting with this one. All right. Let me do a third one. So if I start with it up, the next one in line goes down. So the next one in line is opposite to this, so it flips back up. So it is going to be just like this one. All right. I need to fix carbon 1. All right, so in this one, all right, here carbon 1 is pointing up, right? So in this one, the OH on carbon 1 must be pointing down. Even though it's beta, remember, we are flipping it. So if you start with this version, right, you start with the correct version of beta, and you are flipping it over for the next one, that means this OH comes down, this one goes up. So this OH and carbon 1 in the new one, because we flip it, it must point downwards. And the H is upwards. For the new one, I'm going to draw. All right, so let me go again now. That's not being drawn good. Let's go again. All right. And so again, you're going to, all right, let me just complete over here, OH, H. All right, take off OH from one and H from the next one. All right, so what happened? So this is showing how it is being formed, right? And then we're going to show the finished product. Right, so there you have it. Now, in terms of the level of detail, right? If you get cellulose on the exam, you don't have to see all of these OH groups and so forth. You don't even have to see the CH2OH. So, how do you recognize cellulose? You see this boundary right here, if you notice, it is pointing upwards, but the next one it is pointing downwards, right? Also, you know that this is carbon 6, right? This is carbon 6. It's pointing up, then in the next one it's pointing down. Then in the next one it is pointing up again. That means it is being flipped one after the next. It is rotating 180, one glucose molecule after the next. The only polymer that does that is cellulose. All right. So the only thing I need to look out for is this bond right here. You will see it pointing up, and then the next one it will be pointing down. That is cellulose. Also, you would know that this is carbon 1 and this is carbon 4. So it's 1, 4. And once you see it being pointed up, that's beta. So up and it flip down. All right. So you don't have to get all of these details. Right? But if they ask you to show it, right? Depending on how they phrase it, if they ask you for the full details, right? Or if they say sketch, once they say sketch, you can do it like this, right? But if they want the full details, which they are not going to ask because that takes a lot of time, right? To draw the glucose with everything on it, right? So more than likely, you just draw this structure to show how it is formed. All right. So you have to be able to draw this up here now. You don't have to have all the details, but you must be able to put on the OH groups on carbon 1 and 4 and show how you end up with this here. All right. But I'm going to put on everything since as I'm showing you how to do it. All right. So I'm going to put on back the CH2OH. So once you get comfortable with doing it like this, then you gradually do it with less details because you should be able to recognize it without all of the details on it. Mm. The OH group on the inside, it would point on the outside. This one on the outside would now be right. So that is cellulose. Right, so starch, it is made up of alpha glucose. So unlike cellulose, which is beta, it is alpha glucose. Now, Starch is a mixture now of alpha 1,4 glycosidic bonds and beta 1,6. So how do we get 1,4 and 1,6? Starch, separate starch is the only one now that is made up of two polymers. So, like cellulose, it is made up of glucose, right, which is a monosaccharide. Starch is actually made up of amylose, which on its own is a polymer, and amylopectin. Right? Each of them have 1,4. So that's where the 1,4 comes into play. The bonding in amylose is 1,4. In amylopectin, it's 1,4. But when we're going to join amylose to amylopectin now, that's going to be the 1,6. So let's go. That's a CH2 OH. Right, I'm not going to do it with any CH2 OH because I need to learn now to just see this simplified structure and recognize it. All right, so that's a simplified structure for glucose without any elements being shown other than the oxygen there. Hold on, let me fix something. Let me go here, so look, drop up that. All right. So up here would be amylose, right? So what I'm going to ask you to do, right, for homework, unlike we're in for sucrose and cellulose, I draw them and show you how to remove the water, right? And then I would draw this structure. So I'm drawing it here, which is the finished product. But I wanted to draw them to show me how you would remove the water to end up with this. All right. So this is amylose. That's a section of amylose. Now below that, I am going to put amylopectin now. All right, so as you know, this would be carbon 1, right? And it would have an OH on it. This here is carbon 6, and it would also have an OH on it, right? With two hydrogens, CH2OH. So the 1,6 now So this is amylose, this is amylopectin below it. All right. Now the OH from one of them, and you take H from the next one. So if you should do that, take away the H, right? OH and H from one of them. This carbon now is going to be bonded to this oxygen. All right, so let me just do it now. So this carbon will be bonded to this oxygen. This is carbon 6, and this carbon is carbon 1. So that is how you get 1,6 in starch. All right. So starch is made up of amylose, right, which all have 1,4 links, carbon 1, carbon 4, and amylopectin, which also has 1,4 linkages. But to connect amylose to amylopectin now, it is carbon 1 with carbon 6. So if they ask you for the bonding starch, you say 1,6. But if they ask you to list all of the bonds, it would be 1,4 and 1,6 in starch. But if they just ask you to state it, you use, hold on, why do I have beta there? Sorry, that's not beta, it's alpha. All right, so the main bond we talk about in starch is 1,6. All right, in amylopectin and amylose, it's 1,4. All right. So unless they ask for all of them, you only state 1,6. If you put 1,4, it's wrong. All right, because 1,4 is either amylose or starch. Sorry, it's either amylose or amylopectin. Starch is made up of the two of them. So you have to mention the 1,6 link. All right, and that is how you do starch. And we are going to draw this again for glycogen. So starch and glycogen, it has the same 1,4 and 1,6. However, glycogen is not made up of amylose and amylopectin. All right, glycogen, it has alpha 1,4, just like starch, 1,4 and 1,6. But it's not, but we don't say amylose and amylopectin, all right? Glycogen is made up of glucose units, right? But it is in the same manner, like you see the 1,4 up here, 1,4 below, and connected by 1,6. It's just that we don't call a portion of it amylose and the next portion amylopectin, all right? It's just glycogen made up of glucose units, all right? But if you're asked to draw a portion of it, you do it like this, all right? So it's similar in structure and bond to starch, all right? So in next week's class, because I'm going to get ready for the next one at 2, next week class, well, it will be in this week, yeah, but next class, we're going to, which one did I not? Right, we are finished with the carbohydrates. So we're going to do the lipids and the proteins in terms of their structure. Now we're not finished with carbohydrates. All we did was look at the structure. They also have their properties. So we're going to also, once we don't draw the structure of the lipids and the proteins, we're going to come back now and look at how the structure affects their properties. So for example, glucose is soluble, right? Fructose is soluble as well. Sucrose is used as transport medium in plant. cellulose is insoluble. So we're going to look now at how the structure correlates to those properties. All right. So for now, just practice how to do the drawing. I will post some questions in the classroom based on what we did today. All right. So we're going to wrap it here for today.